Good morning, students. Today we are going to start the next chapter in biochemistry, that is metabolism. So, in the first chapter, we have learned about different types of biomolecules like carbohydrates, proteins, and lipids. So, in this chapter, we are going to see how each of these biomolecules metabolism is breaking down is occurring. Okay, so we can start the chapter. What is metabolism? Metabolism is a very simple basic term that you are learned from very lower classes onwards. What is metabolism? It is the sum total of all chemical reactions in the body as part of metabolism. Okay, as part of breaking down. So metabolism is a sum total of all chemical reactions in body metabolism that results in the liberation of energy for our physiological activity. In metabolism, there are two types of process, anabolism and catabolism. As the name implies, anabolism, ana means making, ana is big. So anabolism is it's a constructive process. So using simple molecules, complex molecules are forming. That is anabolism. In catabolism, cat is small. So catabolism is breaking down process from complex molecules, simple molecules are forming. Then amphibolic, I think this term is somewhat new for all. We need to understand that. Amphibolic. Amphibolic means it's a combination of anabolism and catabolism. If in whatever pathway these two processes occur at the same time, that pathway are called amphibolic. For example, amphibolic pathway example, Krebs cycle that you will learn in this chapter. The Krebs cycle is an amphibolic pathway. In that same pathway itself, we can see both anabolism and catabolism. So, we are going to see the metabolism of first main biomolecule, that is carbohydrate. First point, what are the main features? Several pathways are there for in the metabolism of carbohydrate. So many ways are there. So many different methods are there for breaking down the carbohydrate. And some pathways are catabolic, some may be anabolic. Then some means at this Krebs cycle, it is amphibolic. And all these pathway may start with glucose and or ends in glucose. So which are the features? It has several pathways. It can be catabolic or anabolic and sometimes amphibolic. And also almost all pathways starts with glucose or ends up in glucose. That means it starts with glucose means it ends up in liberating energy. It ends up in glucose means it is an anabolic process, so it conserves the glucose. So important pathways that we need to understand, learn as part of the carbohydrate metabolism are glycolysis, glycogenesis, glycogenolysis, gluconeogenesis, Krebs cycle, and pentose phosphate pathway. All the cycles are important. At least one cycle will be asked for your examination. So for while studying each one, you need to learn how to draw the diagram and to describe each cycle. And also listen which are the enzymes involved, how many ATP, net gain of ATP from each cycle, all those things. So then first you can listen that name glycolysis. Glycolysis means this is a catabolic. The name itself it denotes it's a catabolic. Glucose lysis. Then glycogenesis. Glycogenesis. Ss means synthesis. Means glycogen is synthesizing. So it can be an anabolic process. Then next one see glycogenolysis. Glycogenolysis. Glycogen breakdown of glycogen is again a catabolism. Then gluconeogenesis, again synthesis, anabolic pathway. Gluconeo means non-carbohydrate sources are using for synthesizing glycogen or glucose. That is gluconeogenesis. Then Krebs cycle is an amphibolic, then pentose phosphate pathway. So we can learn each one separately. First one, glycolysis we can leave here. It's somewhat more complex when compared the other three. So after learning these three, we can go to glycolysis. So first we are going to see the glycogenesis. Glycogenesis means glycogen synthesis. Glycogenesis synthesis. Glycogenesis. From the name itself, you can understand what is the basic process going on here. By the mean of this pathway, glycogen can be synthesized. So for one more question, it's also these type questions can come. What is mean by glycogenesis? Then you need to just simply write is the process of synthesizing glycogen from glucose. So glycogenesis, first what is the purpose? Is a synthesis of glycogen from glucose. So we can go and see the process. In this chapter, if we have an idea about some enzymes, it will be more useful. So kinase, to whatever enzymes, if we add that tail, kinase means its function is to add phosphate group to something. If we add phosphorylase at the end of the name of some enzymes, it means that that enzyme can cleave the phosphate out of some substrate. Then if some enzyme have this mutase at the end of its name, that means that enzyme's function is to change the position of some functional group within the substrate. Then phosphatase, again it hydrolyzes the phosphate. So these things we can keep it in mind. So one more thing. While you're studying all these cycles, you need to take a fresh sheet of paper and draw this schematic diagram. And looking on this diagram, you have to study. And with some different color sketch, you have to write the name of enzymes. And you have to write the names of enzymes in one side and all the other process happening along with the pathway on the other side of this arrow. Okay. Also, this is another technique that I often use while I am studying the cycles. So, we first need to look over what are the main steps involved in that pathway and give its important main letters as one by one. So, it will be very easy to memorize. G here glucose along with then the next what change next substrate in that pathway is glucose 6 phosphate. Glucose 6 phosphate again become glucose 1 phosphate. It's very easy to remember. Then glucose 1 phosphate become UDP glucose, then glycogen chain. These are the main steps that is going on this glycogenesis. Then we need to only know which enzyme is acting here, 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 and all what associated side chain process going on here. So if we memorize in this way, I think it will be easy for all what this metabolic pathway. If you create, you write up like this by taking the first letter of main substrate in each step, one by one, one after other as a separate box. It will be more easy, helpful for you to memorize the stepwise sequence. And looking on the name of each step, you can make a guess about the name of enzyme and what all the reaction going on this prior substrate. Okay, let's study the glycogenesis. In this way, we are going to handle each cycle. First, glucose is the main from starting point. Glucose from glucose only we can synthesize glycogen. Mainly. So glucose mainly here, free glucose only can move freely in our blood. So in what form we can store glucose inside the cell, because all the glucose available to the body, if it is the liberated, distributed to the blood without any regulation, it may steep the level of glucose in our blood. So we must have a control. So by converting glucose to this form, glucose 6 phosphate form, we can keep it inside the cell itself without freeing it to the blood. So in this form, it cannot pass freely from inside cell to outside the blood. So wherever we need to convert the glucose to other form, mainly majority of cases we are converting into another form. Then the form is that, listen to that name, glucose 6 phosphate means in the glucose structure 6th position a phosphate group is adding. Phosphate group is adding, that means a kinase. Only kinase can add most oftenly. So here that kinase is hexokinase. So as we know that these are the main substrates. So glucose, after that glucose, it should come glucose 6 phosphate. Glucose 6 phosphate means one phosphate group is coming. For us, one phosphate group is coming, there should be some enzyme, which should be that enzyme a kinase. So this is a 6 carbon molecule, a hexokinase is acting over this glucose to convert this glucose to glucose 6 phosphate. And for this step, energy is required. That energy is provided by one molecule of ATP. Then one molecule of ATP, that phosphate also is taking from ATP. It's hydrolysis occurs, and adenine triphosphate, 3 phosphates are there. From this adenine triphosphate, one phosphate is taking to and adding to this glucose, and this glucose become glucose 6 phosphate. Understand? Only simply this much. Glucose is converted to glucose 6 phosphate with the help of the enzyme. That phosphate group is taken up from ATP. As we got glucose 6 phosphate, then next we need to convert glucose 6 phosphate to glucose 1 phosphate. That means change in the position. That functional group change is always done by a mutase enzyme. So here also one mutase. 6th position, the phosphate in the 6th position is shifted to the first position, first carbon. So it becomes glucose 1 phosphate. Okay, glucose 1 phosphate formed here. Then with this glucose 1 phosphate, it combines with the uridine triphosphate. Uridine triphosphate. Uridine triphosphate. Uridine is there, then 3 phosphate groups are there. Then what is happening here? Here is also one enzyme, UDP glucose pyrophosphorylase. Some transferring also occurring for the phosphate group. So that's why UDP glucose pyrophosphorylase is there. Now that what is happening here? In this step, the product formed is UDP glucose. UDP glucose means uridine diphosphate glucose. Along with this, here a pyrophosphate also formed. PPI, pyrophosphate. How this pyrophosphate is formed? In this pyrophosphate, one P come from this UTP, and one P come from this glucose 1 phosphate. Here one phosphate is present. That phosphate and this 1 phosphate. Then balance what remains here? UDP. That UDP combines with this glucose to form UDP glucose. The enzyme here acting is UDP glucose pyrophosphorylase. I think it's very clear. Then this UDP glucose is just simply adding to a growing chain of glycogen. Growing chain of glycogen means I say what it's a repeating blocks of glucose units one by one, and that way in that way glycogen molecule is forming. Glycogen is a macromolecule composed of monomeric units called glucose. So this UDP glucose, just like we are adding beads to a neck chain, that UDP glucose is adding to a growing chain of glycogen. For that we need to remove that UDP. UDP we are removing, and from this UDP again UTP is replenished by taking one phosphate from a ATP molecule. Okay. Anyway, main thing is this reaction, that UDP glucose, UDP is removed, and the enzyme playing the main role is glycogen synthesis. Glycogen synthesis, what it's doing? It combine this glucose to the growing chain of glycogen with what type of bond? Alpha 1,4 glycosidic linkage, because in the straight chain that bonding between each monomeric glucose unit is 1,4. Alpha 1,4 glycosidic bonds. What is alpha and beta? What's the difference? Everything we have learned in the first chapter of biochemistry. So leave it. In that way, glycogen synthesis is adding glucose monomers. And as the glycogen chain grows to a certain length, then a branching, because side branching may occur. How this side branching is occurring? When this glycogen chain grow to a certain length, then a 7 unit glucose units is detaching from that long chain, and it's forming a side branch for that elongated chain. Using what type of bond? Here it is alpha 1,6 glycosidic linkage with the help of some branching enzyme. This is the glycogenesis. Okay. Next we are going to study about glycogenolysis. Glycogen breakdown, catabolic process. When it occurs, because if our body is facing a depletion in the level of glucose, when the body needs more glucose, what the body will do? Body can't break down the stored glycogen and release glucose. So in that situation, this cycle occurs, glycogenolysis. Low level of glucose triggers this cycles, glycogenolysis. What is its main process? Liver glycogen is converting to glucose 6-phosphate, and that glucose 6-phosphate, when it becomes glucose 6-phosphate, it is very clear that we only need to remove that additional phosphate. If that additional phosphate is removed, then that substrate become free glucose. Free glucose can easily move to the blood and ready to distribute to all our body. So we are going to see that cycle. It's very simple cycle. Here the glycogen is there. First the glycogen is converted. First we need to see which are the main steps. Glycogen, first we need to change to glucose 1-phosphate. Then glucose 1-phosphate, again we need to change to glucose 6-phosphate. Glucose 6-phosphate becoming glucose. So that's the main stages. So we can see which are the enzymes acting, in what way these changes are happening. The conversion of glycogen to glucose 6-phosphate involves two steps. In the first step, each that glycogen reacts with the phosphate with the help of an enzyme glycogen phosphorylase. Because it need make a cleave here and to add this phosphate group to this glucose unit that is freed from that glycogen. So the glucose residue free from the glycogen reacts with the phosphate to form glucose 1 phosphate. The enzyme acting here is glycogen phosphorylase. Okay, and from this glycogen, this glucose 1 phosphate is formed mainly by the cleaving of a main bond. Bond means glycogen means is the it is a polymer of so many monomeric glucose units. Each monomeric glucose units are bonded in a by 1,4 linkage. So at the end of the glycogen chain, one monomeric glucose is linked by using a 1,4 alpha 1,4 glycosidic linkage. So that linkage need to be cleaved. That linkage is actually cleaved by this enzyme. I am just telling what's the what's the main process that is carrying on there. Simply it is only this much. Glycogen is acted upon by the enzyme glycogen phosphorylase, and the freed glucose reacts with the phosphate to form glucose 1 phosphate. Only this much. Glucose 1 phosphate. In the second step, what is happening? This glucose 1 phosphate undergoes isomerization. Means its form is changing to another form. The help of a mutase. What type of mutase? Phosphogluco means you need to think from a phosphogluco mutase. And with the help of that enzyme, what is forming? Glucose 6-phosphate. When it becomes glucose 6-phosphate, you only need to remove one phosphate group in the 6th position of this glucose. Then it becomes free glucose, easy to distribute to the body via blood. So that enzyme is maybe another phosphatase. That its name will be glucose 6-phosphate phosphatase. Then glucose is free. And one more side chain. They say one more what we are saying bypass. Bypass is there. They say this glucose 6-phosphate also have one more fate. It can enter the glycolysis pathway by converting directly to pyruvate. That we will learn later while studying about the glycolysis. Now you have to understand only this machine this way, and it's converting to this glucose. That's about glycogenolysis. Next cycle is gluconeogenesis. Gluconeogenesis is a synthesis, anabolic process. And here glucose we are synthesizing from non-carbohydrate precursor molecule. That is gluconeogenesis. If the question came for 1 mark, you have to write only this much. It is a synthesis of glucose from non-carbohydrate precursor molecules. Okay. Where is the main site? Site is liver. And which are the starting points, starting substrate, which are the substrates, which all things can be the substrate? Pyruvate, lactate, propionate, glucogenic amino acids, any of the Krebs cycle intermediates. That Krebs cycle everything we will learn later. So you just hear by this much only now. Krebs cycle intermediates. All these can act as the starting point for gluconeogenesis. All these are non-carbohydrate substrate also. And also from galactose and fructose also glucose can be formed. So we are going to see the pathway, and as we are all, every time in what way we are memorizing all the pathways in our mind. So in that way we are going to start here also. Lactate, main what steps in this pathway are lactate, then pyruvate become oxaloacetate need to change to phosphoenol pyruvate, then phosphoenol pyruvate become fructose 1,6 bisphosphate, fructose 1,6 bisphosphate, again change to fructose 6 phosphate, fructose 6 phosphate becoming glucose 6 phosphate, glucose 6 phosphate. When it becomes glucose 6 phosphate, it can very easily converted to glucose. These are the main steps. So we are going to see which are the enzymes acting each pathway and what are the associated reactions. First, lactate is converting to pyruvate. Okay, lactate is converted to pyruvate with the help of what enzyme? Lactate dehydrogenase, a dehydrogenation process. Lactate water molecule is losing, and as a result pyruvate is formed. Then this pyruvate is mainly converting to phosphoenol pyruvate through a two-step process. Pyruvate is converting to phosphoenol pyruvate through a two-step process. The first step, pyruvate combines with carbon dioxide to form oxaloacetate. Pyruvate combines with carbon dioxide to form oxaloacetate, and the energy required for the steps is obtained by the hydrolysis of ATP. So one molecule of ATP is consuming here for getting the sufficient energy for the proceeding of this step. Then that enzyme catalyzing this reaction is pyruvate, carbon dioxide is here. So carboxylase, pyruvate carboxylase. Okay, then as a result what is formed? Oxaloacetate is formed here. Then the oxaloacetate we need to convert to phosphoenol pyruvate. That step is catalyzed by the enzyme. We can start with the substrate's name. Phosphoenol pyruvate, then again carboxylase. Phosphoenol pyruvate carboxylase, another reaction. Here also energy is required. All the carboxylase reaction we need energy. The energy is provided by GTP. One ATP is used and one GTP again is used. Then the phosphoenol pyruvate is again then converted into fructose 1,6 bisphosphate. It is a four-step process, and all these four steps are present in glycolysis also, but that steps present in glycolysis, that step are a reversal of these steps that present in gluconeogenesis. What that four steps that occur for converting phosphoenol pyruvate to fructose 1,6 bisphosphate. That four steps actually it is a reversal of the four steps present in glycolysis. That means these four steps as such is present in glycolysis also, but its direction in what way? In glycolysis, if it acts in forward direction, in gluconeogenesis the same step acts in backward reaction, backward direction. That is the difference. Then in that way means the phosphoenol pyruvate as a result of four-step conversion it becomes fructose 1,6 biphosphate. This fructose 1,6 biphosphate is converted to fructose 6 phosphate. That means one phosphate is removed. With the help of the enzyme fructose 1,6 bisphosphatase. fructose 1,6 bisphosphatase. And as the fructose 6-phosphate is formed here, then what is the next step? We need to convert the fructose 6-phosphate to glucose 6-phosphate. How glucose 6-phosphate is formed from fructose 6-phosphate? How it is formed? With the help of enzyme glucose 6-phosphate isomerization reaction. The help of isomerization reaction, fructose 6-phosphate is converted to glucose 6-phosphate. When it becomes glucose 6-phosphate, very easy. It can be converted to glucose with the help of glucose 6-phosphate phosphorylase, maybe that ends. In that way, it is converted to glucose. Where we finish today's class. Thank you.